And now to introduce our first keynote speaker for today, Dr. Alex Kwan. Dr. Kwan is an associate professor at the Meinig School of Biomedical Engineering at Cornell University. He is broadly interested in the neurobiology of drugs for treating depression. His work has revealed how compounds such as ketamine and psychedelics impact dendrites and cortical microcircuits to enhance neuroplasticity. Alex supports open science by posting preprints and sharing data and code for all labs' recent studies. He received a B.A.Sc in engineering physics at Simon Fraser University in 2003 and a Ph.D. in applied physics at Cornell University in 2009. From 2013 to 2022, he was a faculty member in the Department of Psychiatry at Yale School of Medicine. Outside of the lab, Alex enjoys drinking a good cup of espresso and rock climbing with his kids. The title of his presentation today is Psychedelic Drug Action on Neural Circuits. Please take it away, Dr. Kwan.
Okay, yeah, that was a great introduction, and let me try to square my screen here. Okay. Yeah, so thank you for this opportunity from the IMP for me to share some of my lab's recent research. I'll be telling you about some of our work on understanding psychedelic drug action on neural circuits. So I know that most of you are very familiar with psychedelics and psychedelic research, but let me still go through some introductory slide to give you a sense on how I think about this field and what I think are some of the interesting opportunities. So my lab, we're very much focused on psilocybin, and psilocybin is the naturally occurring compound that can be found in a variety of psilocybe mushroom, and there are about more than 200 species of these psilocybe mushroom. That includes psilocybe lineaformum, which is shown here on the top left. This is actually a species that can be found in the Pacific Northwest, including in Vancouver, British Columbia, where I grew up. There's also a psilocybe mexicana, which is probably the most famous species, I would say, that contains psilocybin. And this is the species that was actually in the 1950s, Albert Hofmann used to isolate chemically the active ingredient, which turned out to be psilocybin and psilocin. And finally, on the right is a beautiful example. This is a psilocybe subaeruginosa, which is a species that can be found in Australia and New Zealand. So these so-called magic mushrooms can be found all over the world, and I think contribute to the rich history of research and studies in these areas. And I think most people are most familiar with the acute behavioral effect of these compounds. Which to illustrate the acute effect here, I took an excerpt from a study that was done in Denmark. Here we have one adult female participant that had been administered psilocybin, and this is what she drew as well as write about the experience. So on the left, you can see what she drew, very colorful, I would describe as ribbon-like imagery. And on the right, she wrote, "My inner vision became the universe, filled with colorful waterfalls, glistening like stars not bound by gravity. They floated in the air and folded around each other, totally quiet. The streams were infinite and beautiful." So I think I picked this passage because it illustrates how visual some of these effects are. It's a very colorful senses that is being induced. And moreover, there's also an emotional aspect. Some people would characterize this as connectedness with the world or connectedness with other people, or even a mystical-like type of experience. So these effects are acute in the sense that they last for anywhere from 4 to 6 hours after oral administration of psilocybin. In other kind of route of administration, it could be even more faster, but they tend to be, again, happen within several hours and then afterwards would dissipate. But what's exciting about the field of psychedelics and also in general, the excitement now of the reemergence of this research is because of the potential of therapy. And I think among the different clinical trials, some of the most advanced trials now has to do with psilocybin and depression. So now there has been a number of these phase 2 clinical trials done at different places, including Imperial College London, Johns Hopkins, at Zurich, also by Compass Pathway, as well as the nonprofit Usona. Here I show one graph that was from the Usona study. These are phase 2 clinical trials, so not too big sample size, but there are a decent amount of participants. So you can see on the order of 50 people for the placebo arm, we received niacin, as well as 50 people that received psilocybin. And what you can see is a clear reduction in the depression score when you compare the two arms for psilocybin. And what's most striking is how long-lasting this effect can be. Here, 43 days follow-up after the initial single-dose administration. You can see that substantial reduction in the symptom, in this case for a major depressive disorder. So because of some of these clinical efficacy that's been shown now in the ongoing clinical trial, there has been a lot of attention in the media for psychedelic and potential for psychedelic medicine. New York Times wrote, the psychedelic revolution is coming. Psychiatry may never be the same. Newsweek have this cover article with the magic mushroom and proclaim it as a new treatment for depression. But I think along with many of the media attention, there are also a lot of hype. So you may have also seen, along with these headlines, Prince Harry talked about how he used psilocybin to deal with grief. This is on 60 Minutes on CBS. You have headlines such as this one from the Wall Street Journal: the working woman's newest life hack is magic mushrooms. And then finally, one of my favorite, NPR, that says National Park Service wants humans to stop licking this toad. Of course, they're referring to the Sonoran Desert Toad and 5-MeO-DMT. So I think as scientists, as researcher, we really have a duty to do rigorous study and to be able to spread information to people, the appropriate information, to help people in the general public to be able to separate sort of facts from fiction and sort of the realistic expectation from some of the hype. So given this, what do we know about how psychedelic work? I would say not a lot in wide agreement. Among the things that I think we can all agree on, I would say that psilocybin is known to be a prodrug that can be metabolized into psilocin. And then psilocin, we know the chemical structure very closely related to serotonin. And here you can clearly see how the two structure both have the tryptamine backbone with two slight modification. But unsurprisingly, as a result, psilocin also bind with high affinity to a variety of serotonin receptor. So here you see some of the receptor that express highly in the brain: serotonin 1A receptor, 2A receptor, 2C receptor, binding at order of 10 nanomolar level of affinity. And I would say these basic properties are quite established for psilocybin. But beyond this, and I think which is what I want to have you as a take-home message for today, is that beyond some of the chemical aspect, I don't think we know quite a lot about what psilocybin does in the brain. Much of how it produces the behavioral effects and the changes to how we go about our everyday life is still unknown. So I drew here as a black box. And what I hope you convinced you today through this presentation is that many of these effects, I think, could be enlightened, and we could gain a lot of insight by trying to understand what the drug do to the brain at the level of neural circuits. And I say this because at the end of the day, neurons and the firing of neurons, and also how these neurons can communicate with each other in the level of circuits, is ultimately what produces the behavior. So understand how drug can influence the brain at the circuit level, I think will tell us a lot about why the drug can elicit these wide ranging of interesting acute as well as long-lasting behavioral changes. So today's presentation, I'll give you some example from my own lab about some of the projects that we do to try to address these questions. And I think we're making some good progress. And I think it's also a very exciting place to be because you can apply a lot of cutting edge systems neuroscience type of method to answer these questions. And I expect more work will come from not only my lab, but also from the field in the next 5 years, where we can make a lot of progress in this topic. In terms of impact, take a broader step back. I think understanding the drug action on neural circuit could also have a lot of meaning for drug development, where now there's a lot of effort to understand other types of psychedelics beyond psilocybin, but also psychedelic-like drug, for example, agonists that may not cause hallucinogenic action. Neural circuit could be a common substrate to understand whether the drugs have therapeutic value or not. And then finally, I think because of the similarity of serotonin, I'm also very excited about how understanding psychedelic could inform something fundamental about neuromodulation in the brain, particularly in the relevance to serotonin biology. So given that as a background, today I'll tell you a presentation that has 3 parts. The first part has to do with a research that lab has published already a few years ago. This will be short, but has to do with the fact that psilocybin's action on dendritic spines. For those of you who may not be as familiar with basic neurobiology, this is a drawing of a pyramidal cell. You can see the dendrites coming out. The little protrusions are where it receives the input, which is the dendritic spine. So I'll go into more detail there. And then the second and third part of the presentation will be taking a step back, beyond the cell level and try to understand going to other brain areas and broader in the brain to try to understand what is the drug's impact on neurocircuits. And before I go further, though, this is the people that actually do the work in the lab. I have a lab where we have on the order of 10 to 12 people, mixed with student and postdoc. I'll try to highlight their individual contribution as I go on. And we're also funded by a number of sources, including particularly the National Institute of Mental Health, which fund the bulk of this work, as well as some support from Brain Initiative and One Mind. So when we first received psilocybin and started studying it, this was, I would say, about now 5 to 6 years ago in the lab. We were not quite sure what we're getting into. The lab had a lot of experience studying ketamine, but psilocybin was new. So graduate student Clara Liao and postbac Ian Gregg were some of the first to work on this compound in the lab. And we did the classic behavioral assays that many people in the field ran, which is the head twitch response assay. So here on the left is a mouse that received a single dose of psilocybin at 1 milligram per kilogram. On the right is a mouse that received saline. We have an overhead camera that captured the movement of the mouse, and hopefully you can see the head twitch right here. You can see the head has this lateral movement. This is really the so-called head twitch response. It's actually much faster in real life. This video is slowed down 6 times so you can see the head movement. So we were very happy to see this when we got the compound. I remember the whole lab was very excited, and many of us beyond Clara and Ian, myself as well, it was huddled around the lab bench and just want to see the head twitch for ourselves. But what Clara and Ian did was beyond just initially seeing it, was also did a very systematic and dose response characterization with over 80 mice, both male and female animal. And what they found is that, consistent with earlier work, at about 1 milligram per kilogram, and it's when this drug becomes psychoactive in mice, for C57 mice. So you can see here at lower doses, the effect is not detectable compared to saline control, but at 1 milligram per kilogram, you see this inflection point where it causes a lot of head twitch response. And this will be the dose of psilocybin that we'll use for all of the subsequent studies. A lot of our early work was informed by work done by a colleague of mine at Yale, Ron Duman, who was a great scientist as well as a great mentor who sadly passed away several years ago. But Ron and other people, most notably Lisa Montega, has a so-called neurotrophic model, which says that drugs like ketamine, which is a fast-acting antidepressant, may work by changing excitatory synapses in the brain. So here you can see a schematic of one of Ron's reviews showing that ketamine can potentiate the synapse either by enlarging the synapse or perhaps growing new excitatory synapse. And so one of the questions that we had early on was whether psilocybin might do something similar to some of these more established antidepressant compounds. So to do that, we did imaging. This is a method that we use a lot in the lab, and I'll tell you a little bit about it, and I'll keep referring to it later. But a lot of the lab, we work on head fixed mice. So these are typically awake mice, but their head is fixated for cellular resolution imaging with two-photon microscopy. We're very interested in the medial frontal area, mostly because of its role in depression, but also in antidepressant action. This corresponds to the cingulate and premotor region in the mouse. We label the neuron with fluorescent protein. So this could be either, in this case, with the thyroid GFP-M mouse, where the pyramidal neuron have green fluorescent protein, or in other cases, we use virus to express fluorescent protein. And finally, we replace the skull with a piece of glass window for optical transmission and for imaging. So if you come to the lab, this is an image that you might see. Here we're in the two-photon microscope going into the brain. You can see the imaging of the dendrite. This is a dendritic tree, the apical dendritic tuft. You don't see the cell body because they're very deep. These are deep layer 5 pyramidal cell. But hopefully you can see the dendritic branches where these neurons receive the input, and also the little protrusions along the dendrites. which are the dendritic spines where the excitatory inputs are located. So this work was published. I'll go through quickly, but Ling Xiao was a postdoc who led this work. She imaged these spines day after day, every 2 days for multiple sessions. You can see the quality of the data allow us to track the dendritic spines over time. And what she found is that a single dose of psilocybin can lead to a long-lasting increase in the number of these dendritic spines, which is shown on the left here as spine density. She also found that the size of these spines can also change. So you can see an elevation in the head width, the size of these spines, although it has a different time course, and you can see that it returned to baseline in day 34. Again, just emphasizing the time scale of this, I think what's exciting for us, but also for people in the field, is how long-lasting these structural changes also seem to be. As far as I know, there's not so many things in the brain where after the drug has such a long-lasting effect. And it is thought that some of these structural changes as a result may underpin the behavioral changes that is also quite long-lasting and seen in the clinical trial. Some people also ask whether the structural changes lead to functional changes. So Pasha Vivodian is an MD PhD student in the lab, and he recorded slice electrophysiology. And this is a recording with miniaturized EPSC. And basically, he found that some of these structural changes correspond to also an increase in excitatory neurotransmission in the brain. So there's also functional consequences along with the structural changes. Again, I don't want to go into all the detail about this study, which you can read yourself in the paper. But we are excited about this finding because we thought that some of the structural changes could have biological significance. Many people have talked about synaptic atrophy in the context of depression. So increase the number of dendritic spine could reverse some of those abnormalities. Another way we like to think about this is that, and I think these can happen in parallel, not necessarily either or, is that increasing spine density could also facilitate the integration of new experiences. The reason is that you can also think about when do the brain tend to grow new spines is when the animal tend to learn new things. So there are a couple of pioneering studies here showing that when a mouse start to learn a new motor task, then you can see new dendritic spines growing in the motor cortex. So we think that by inducing structural plasticity, psilocybin could also facilitate integration of experiences. So that, I think, has been quite exciting as a first study in this field, and also quite foundational to how we think about these problems and inform us to some of the current work that has been happening in the lab. And the specific question that has motivated the follow-up study in the lab is that we see psychedelic evoke plasticity, which is very exciting, but which brain circuits are they involved in? I think there's a general recognition that plasticity itself might not be explaining everything, because one could also say that learning can also induce plasticity. Even by virtue of just listening to this talk, you probably also have a new experience, and your brain circuit and wiring would also be changing. And one could also argue that other drugs, for example, drugs of abuse, or basically any kind of psychoactive drug, also influences neuronal function and therefore induces plasticity. So I think just the fact of saying psychedelic induces plasticity, while it's interesting, much more interesting, I think important, is to understand which are the cell types and which are the brain circuits that are involved that make psychedelic-induced plasticity special and unique. So in answering this question and trying to pin down on the brain circuits that are involved, I like to bring this analogy of solving a jigsaw puzzle. I think there are multiple approaches that are possible. If you're thinking about solving a jigsaw puzzle and you have one piece that you know is the starting correct piece, for example, in our case, the starting piece would be the neuron growth spines. One could take a strategy where you can find other pieces that have similar color. So this is a very fast way of doing things, but maybe not necessarily always correct. Another way to do things is to find pieces that connect and lock in one piece after the other, which is laborious, but you know that you're slowly completing the picture. And I think both of these approaches have value, and I view these approaches of solving the puzzle as, on the left, is a high throughput mapping type of approach, which is essential, in fact needed, and the only way if you want to do drug development and find new compounds. On the right, I would say is more of a biologist approach, where we're trying to understand the biology and the detailed biological mechanism that fit under those findings. So I'll tell you a study in my lab that leverage actually both approaches, that take both approaches. I'll first tell you one CFOS imaging study, the second part of this talk, where we took a high throughput mapping approach, and then I'll end using most of the time actually to talk about the detailed circuit tracing, where we find some very interesting cell type specific circuits that are involved in psychedelic drug action. For the high throughput approach, we leverage the fact that when there's a synapse potentiation, that tend to be preceded by neuroactivation, calcium influx, and then also immediate early gene expression. So these are genes that are quite well known. For example, CFOS, ARC, MPAS4. These are genes that's related when a cell needs to respond to external stimulus, need to synthesize new protein in order to adapt to the new environment. So MD PhD student Pasha Davodian did a CFOS imaging study. So what he did is that he would inject mice with psilocybin or ketamine or saline, collect the brain, clear the brain, do immunolabeling for CFOS, and then use light sheet microscopy to image the CFOS expression in the brain. Here is one example of the image that he could get. This is one slice. So he would apply the light sheet. This is one plane. And for the whole brain, he would image on the order of, I believe, 40 to 60 planes. But you can see actually way more than 40 to 60 planes, I think, because it's cellular resolution, probably on the order of hundreds of planes. But what you can see, though, is that at this resolution, the clear cellular resolution CFOS immunolabeling. On the right is an inset of the cortex, where you can see the puncta correspond to the CFOS positive cell acquired using the light sheet fluorescence microscopy. Here's the one slide that I showed that you cannot see, but I just want to illustrate how much big data can be generated from this approach. Because we're imaging the whole brain, we can segment it into 316 different brain regions as defined by the Allen Brain Atlas, cortical region, thalamic region, hypothalamic region, brainstem region, and we can ask, compared to saline, how ketamine and psilocybin induces different CFOS expression changes. Some of the places have a common and shared response. For example, cingulate cortex, ACAD, the dorsal area of the anterior cingulate, the ventral portion of it, prelimbic region, visual cortex, retrosphenial cortex. There's a number of regions where both of these drugs increases the amount of CFOS positive cell. Some regions are psilocybin only, which we find quite interesting. Not many, but there are a few of these. For example, the anterior insular cortex, PAG, and dorsal raphe. Dorsal raphe was particularly great to see because there's a lot of historical work from George Akaginian and Barry Jacobs showing that psychedelic can induce changes in activity, specifically suppressing the firing rate of dorsal raphe neurons. And then there are some regions have ketamine-only response, for example, infralimbic cortex, CA1, CA3. So yeah, again, I don't know if you can see all the regions, but if you have any kind of favorite region that you want to study and try to understand, I encourage you to go check out this paper to see if they may be responsive to the drug of interest. What's exciting for us, though, is that with this large data set, we can also now correlate with other type of big data to explore what might be driving some of these drug-evoked gene expression changes. This includes using bioinformatics data, which I'll give one example, machine learning method, I'll also give one example, and hopefully in the future, I'm optimistic about maybe combining with some human neuroimaging data to get insights on what might be driving the brain's effect along the whole brain. For bioinformatics, just as one example, what we've done is use our CFOS whole brain data, but also take the in situ hybridization data from Allen Institute to ask the question, what might be the genes that might be important for driving these drug response? So what we can do is take how each gene are expressed across the whole brain. And in the mouse genome, there's 20,000 plus genes that's been mapped this way. And for each of these 20,000 genes, we can try to correlate with the drug-evoked CFOS pattern that we see. So that lead to an analysis such as this, where we look at specifically serotonin and dopamine receptor as candidate genes. But let me take some time to go through this plot. The gray histogram that you see is the correlation between all 20,000 or so genes with the CFOS expression pattern on a region by region basis. So that's basically all the genes that's possible. And then each of the dotted line, the vertical line, are the candidate genes that we were interested in. So what you can see here is that the serotonin 2A receptor correlate actually quite well. So if a region expresses the 2A receptor, they tend to have higher CFOS response. So the 2A receptor at 70-something percentile seems to have a good link with the drug response, whereas some of the other serotonin receptor and dopamine receptor does not seem to relate so much with the drug action. When we dig a little further, we also tested glutamate receptors. And what's interesting is that many glutamate receptors, AMPA subunits, as well as NMDA receptor subunits, actually correlate even higher than the serotonin 2A receptors. So whether a region has some of these glutamate receptor transcripts relate very much to whether they respond to the drug. I think going in, we didn't know, but I think after doing an analysis, we think that this does make sense because these drugs can induce formation of dendritic spines, which would contain some of the excitatory synapses. So the fact that I think some of these glutamate receptors correlate with drug action perhaps is not unexpected, but I think it's very good to see. We've also, in the paper, I won't show it here, correlate with different types of cell types. And obviously, you can also look at many of the other genes that might be possible as a kind of exploratory analysis to ask what might be some of the molecular pathways that are involved. Beyond bioinformatics, we also did machine learning. This is a bit of a busy slide, but this is work led by MN student Farid Abuhab together with Pasha Davodian, where instead of just 2 drugs now, we did 8 different drugs. So psilocybin, 5-methoxy-DMT, 6-fluoro-DT, which is an older known non-hallucinogenic 2A agonist. We also included ketamine, MDMA, and then fluoxetine, acute administration as well as chronic administration, and saline. This work we recently put on bioRxiv. But here the question was, can we use these methods to actually screen and classify drug? Can we ask the computer to predict which drug the mouse had been administered based on purely just the CFOS map that is given? And what's shown on the right is that the computer can actually do this to fairly high accuracy. So you can see the predicted label and the true label on what those drugs are. And on average, the computer can get about 70% accuracy on guessing the right drug. Since there's 8 drugs, the chance level would be about 12.5%. So the computer is doing way above the chance level in using the brain activation pattern in order to predict what drug has been given. Our hope for this kind of research is that these drugs are all well known, but if you have a new drug that you don't know, you can potentially give it to the animal, map its CFOS expression pattern, and then be able to tell which drug might it be close to in the wide range of potential drug space. Okay, so that was the second part of this talk where I tell you about one high throughput method, which I think has a lot of relevance in terms of drug development, but also allow us to generate different kinds of hypotheses about how the drug will work. And then for the final part of the talk, I'll tell you about some study where we look at specific cell type and try to understand the biological mechanisms. One of the things that we knew from the early study is that pyramidal cells can grow dendritic spines, but it's also quite well known that they're not just one type of pyramidal cell, but in fact, the brain has several types of these excitatory cells, and they're subtypes of pyramidal neurons. This is a review from the lab of Gordon Shepherd at Northwestern, where he highlights some of the major subtypes of these excitatory cell. You have the intratelocephalic cell, the so-called IT neurons, pyramidal tract neuron, which is the PT cell, as well as cortical thalamic neurons, the CT cells. You can see that, first of all, these cells distribute differently in the cortex. So the IT cell, you can find them in almost all layers in the cortex, except for layer 1. But all the way from layer 2 to layer 6, you can find populations of IT cells, whereas PT cells are quite different, and they're localized to deep layer 5. And finally, most of the cortical thalamic neurons are located in layer 6. Beyond the cortical distribution, there are also distinct differences in where these cells project to. So they output a different brain area. You can see the IT neuron on the top. They innervate heavily the basal ganglia and the striatum, as well as outer cortical regions, whereas the PT cell does not or have very few cortical connection. You can see they dive deep right away, going into and through the basal ganglia. Some of it go to the thalamus, some of it go to the pons, and some of it even down to the spinal cord. So we think that it will be very important to understand how these different excitatory cell type might respond to psychedelic, because it will inform us what are some of the output pathways that are important. We know the frontal cortex is important, but how does it mediate some of these actions downstream? What are some of the downstream brain region and output targets that are involved in a drug action? So we first did an exploratory analysis using the single-cell RNA-seq database from Allen Institute to see what kind of receptor these cell expresses. And what we find is that both these IT and PT subtype actually express a good amount of the serotonin 2A receptor, which we think are quite important for the drug action. So here you can see on the vertical axis the different cell types. The IT cell, again, have different layers. PT cell is just in layer 5, and the top is the cortical thalamic cell. The cortical thalamic neuron doesn't have that much serotonin receptor expression, but hopefully you can see that for both the IT and the PT, they express a lot of this HTR2A transcript, which is the one that encodes the serotonin 2A receptors. So both of these cell types seems like it could respond to the drug quite vigorously. So to study these cell type, then we did a variety of study, including labeling them, manipulating them, and also imaging them. So this is work led by postdoc Ling Xiao. One of the ways that she used to target these cells is through viral strategy. So here you see that Ling Xiao inject the virus AAV retro-Cre into the pons, and then also AAV flex GFP into the frontal cortex. This allow her to label the specific population of PT cell. And on the right, you see the histology showing the expected lamina distribution for these cells. And then for the PT neuron, so for the IT neurons, that was for the PT neurons. For the IT neuron, you can see that using a similar but different targeting, here she injected the contralateral striatum, which allows her to now label the IT neurons specifically. So using these viral strategies, she can express fluorescent proteins for imaging in these different cell types. The first thing that she did was to repeat the spine imaging study, looking at structural changes and whether some of the dendritic spine growth could be different between these cell types. Here you can see that she imaged twice before, give the animal psilocybin or saline, and then image more times afterwards. A lot of people also ask her and also ask me about how long this lasts. So we added one more time point now 2 months later. The previous study, we only did 1 month later. And this is about as long as we can keep the animal and the window still clear for imaging. But you can see that at day 65, in example here, you can still see the spines quite clearly. And what Ling Xiao found is that in the PT subset, she replicated her early result. You can see the increase in spine density that increases quite dramatically after the single dose of psilocybin. It was sustained all the way out to day 35 and then into day 65, suggesting that these density increase and number increase actually is sustained even longer than we previously thought. She also saw the spine size change increase, although this also go back to baseline over longer time points, again, similar to previously. And so now next we want to look at the IT cell and see if there are some differences. What was a little bit surprising to us and maybe initially disappointing was that the IT cell also turned out to have increase in spine density. We're really hoping for some cell type difference in the density. But instead, what we did find a cell type specific difference is on the spine size change. So what I'm showing you here is that in the spine density, both cell type show an elevated spine density increase, but the PT cell preferentially show an increase in spine size. You can see we did a mixed model with a treatment and cell type as a factor. There's a significant difference across the cell type in how they respond to psilocybin in terms of the change in the dendritic spine size. So again, just reiterating, both of them, the spine grow, but in the PT's case, in addition to just growing new spines, the spines also got bigger, whereas the IT cell, that's much less prominent. So this, to recap just this one part, there's structural remodeling both cell types, both of them grow new spines, but the PT cell seems to uniquely have these larger dendritic spines that appear after psilocybin administration. One of the key questions then is, do these things mean anything actually for behavior? This is also one thing that we didn't quite test last time. Are these cell types involved in psilocybin's behavioral action? To answer this question, Ling Xiao used chemogenetics. So here, to target these neurons now, she uses transgenic mice. Using the FESF2 Cre-ER mice, she injected virus to express channel rhodopsin actuators to express them in PT neurons. And then using the plexin D1 mice, she expresses a chemogenetic actuator in the IT neurons, both of these bilaterally in the medial frontal cortical areas. The experiments look like this. On the left is the learned helplessness test, where we subject the mouse to stress induction for 2 sessions, including inescapable foot shock. And then afterwards, give the mouse DZZ, which would activate the chemogenetic system. This either silences the PT cell or IT cell. While those cells are taken offline, we give the mouse saline or psilocybin. And then finally, we would test the effect of that initial stress in terms of inducing learned helplessness. So again, the goal is to take some of these cell types offline and see if the drug can still be active and behaviorally beneficial. So first, we again replicated some of our earlier finding where for the control case, where they don't have the chemogenetic ligand, but they just have a M-cherry control fluorophore express, we find that the animal fail to escape less when they have psilocybin. This means that they're less sensitive to the stress. So here you can see that the saline group failed to escape in this learned helplessness test, which suggests that they're very sensitive to the stress. But in the psilocybin case, the failure rate has decreased. And what's striking is that now when we shut down the PT cell, we find that the drug is no longer active. So you can see the group that receives psilocybin fail just as much as the saline group, suggesting that normal PT neuron function in the medial frontal cortex is important for psilocybin effect on the stress-related assay. Ling Xiao also tried this on the IT neuron, and what she found is that in the IT neuron, there's still a decrease in their failure even when she shut down the IT neuron, suggesting that here we have a dissociation where the PT cells are quite important for the behavior action of the drug, but the IT cell, you can take them offline, but psilocybin could still be active and still produce the behavioral changes on the day after when the drug is off the system. We also tested, in addition to stress assay, the acute changes, in this case, a head-twitch response. What is shown here is that no matter whether you shut down the PT cell or the IT cell, the head-twitch response remained the same, suggesting that even though the PT cells are important for the long-term stress effects, short-term, neither of these cell types contribute to the head twitch. So I won't show you about all the behavioral tests that we've done, but beyond these two, we've also done tail suspension, we've also done fear extinction, and the result is largely the same. We're showing that the PT cell is quite important for some of the long-term behavioral changes. So up to this point, now what I've shown you is that the PT cell, not only do they have cell type specific remodeling, but they also seem to be specifically involved in the relief of stress-induced behavior for psilocybin. And neither of these cell types are involved in the head-twitch response. At this point, we want to know what is so special about psilocybin drug action on PT neuron. Is there something unusual about how they respond to the drug when the drug is administered that endow them with these different cell type specific long-term effects? To answer this question, we did a number of studies. One is calcium imaging, looking at calcium influx at the dendrite of these neurons where the spines are growing. This shows a two-photon image of calcium influx in the dendritic segments. Some of the events can be quite widespread, some of them quite local, but you can see the calcium signals in these dendrite is quite a rich signal. What Ling Xiao found, and also with MD PhD student Neil Savalia, is that acutely after psilocybin, we can image the calcium transient using GCAM6F, and the PT cell show an elevated calcium transient increase. So these cells also specifically have a more calcium signaling happening after psilocybin administration. Comparatively, the IT cell did not have any kind of changes in the calcium signaling during this period. One final thing that we did for this is we also did some electrophysiology to measure the firing rate of these neurons. So here we did not do imaging, but instead we stick an electrode here using neuropixel probe into the medial frontal cortex and recorded firing activity from these cells. Afterwards, you use optogenetics, optotagging strategy to see what cell type we recorded from. And for PT neuron in the FSF2 mice, you can see that when we do the optotagging and shine light, this cell respond by spiking, suggesting that this cell does express tranodopsin, and then we can confidently assign this cell as a PT cell. And what the electrophysiology show, and this was work by Pasha, is that a subset of the PT cell respond quite vigorously to psilocybin. You can see in general, the saline panel, psilocybin panel, in general, these cells after psilocybin has somewhat of a heterogeneous response, some increase, some decrease. But I think, again, what you can see quite clearly is that about 20% of the PT cell, in addition to some of that heterogeneity, respond fairly vigorously with a high increase in firing rate following psilocybin, which was quite distinct and I think stood out from some of these studies. So to summarize this last part in terms of the results, what I show you is a cell type specific circuit that I think is quite important for psilocybin's drug action. We found that acutely within hours, the PT neurons show increase up at the dendrite in terms of calcium signaling. They show increased firing at the cell body, which I think later on lead to some of the cell type specific structural remodeling changes. And also perhaps the reason why these cells also seems to be important for mediating stress-related behavioral effects of the drug. So I think this study, as any good study do, I think it lead to, for us at least, it lead to more questions, I think, than answer. I think it's a very important finding, but it lead to additional questions and unanswered, I think, problems. One is, yes, okay, the PT cell are important, but why are the PT cells then preferentially excited by psilocybin? Why are their dendrites showing their calcium transient increase, and why are they firing more? One possibility is, I think, because maybe the 2A receptor act differently on the cell type. So at the very get-go, if you remember, I showed you that both PT and IT cell express the 2A receptor. So just the presence alone of the receptor, I don't think is different between the cell type. If anything, the IT cell have more 2A receptors. But there could be other differences, for example, of how the downstream signaling is mediated, the GQ and beta arrestin signaling, or maybe how these receptors are distributed within the PT neurons that make the PT neuron the preferential responder. Another possibility that I'm in favor of, and we're testing right now quite actively in the lab, is maybe it's not the cell itself, but the cortical microcircuitry involved and the other cells that's within the cortex that lead to the PT preferential effects. And what I'm talking about is the range of GABAergic interneuron, as well as other excitatory cell, for example, the layer 6B cell in the cortex. These cells can modulate microcircuit activity in different ways that could lead to preferential circuit activation by the drug. And following this vein, we have now started looking at different type of GABAergic neurons. We can do similar kind of analysis to ask what are some of the serotonin receptors expressed in these interneurons. Indeed, the interneuron express different kinds of serotonin receptors, so they should respond to the drug and alter the pyramidal circuit activity as well, indirectly through the GABAergic neuron action. As a start, I'll show you some data with the PV cell, which, as you see here, express a 2A receptor, so we would expect their firing to increase after psilocybin. Indeed, when Pasha do these recording, now instead of optotagging the excitatory cell, he optotagged the PV interneuron and showed that after psilocybin, these cells does increase their firing quite substantially compared to pre-drug condition. So this is, again, an area I think we're quite excited about, looking at going beyond the excitatory cell, looking at broader circuits, also maybe looking at some of the neurons that connect to other brain regions that would respond to drug action. So yeah, just a final 3 slides now to really sum up. I do want to mention that so far all I tell you, even though I think these are important circuit mechanism, has to pertain to psilocin and really the action of psilocybin. Psychedelics is broader than that. There are different subclasses, and then within it, other compounds like LSD, 5-methoxy-DMT, for the tryptamines, but also muscarinin, DOI, and so forth. So I think one has to consider the potential varying action between these different drugs. What works for psilocin may not be the same for different things, for example, like for LSD. So I think that's something to keep in mind. And if you're interested in all the psychedelics, I also encourage you to attend a Gordon conference next year. I think there's not a whole lot of scientific conferences for discussing psychedelic research. This is something that I'm quite excited about, where several colleagues and I have now established. This is the inaugural conference under the Gordon Research Conference series to discuss psychedelic research. And we have an excellent lineup of speakers. This will happen next year in July. And to sum up today's talk, initially I tell you about psilocybin and why studying its action is important, thinking about mental health. I tell you about our earlier work looking at structural remodeling changes. Then I tell you about CFOS imaging as a way, for example, for understanding plasticity-related gene expression and for drug development. And then finally, I told you a specific pyramidal cell subtype circuit that we think is quite important for the psychedelic drug action for psilocybin. So with that, yeah, I hope you enjoyed this presentation, and I'd be happy to answer any questions.
Thank you, Dr. Kwan. That was super fascinating. This is my my wheelhouse here, so I was really fascinated throughout the talk. We do have several questions in the Q&A, so we can just go down the list. Someone asked, is the 5-HT2A receptor expressed in Thywan neurons?
Yeah, so I'm glad I see the list now. I'm glad you asked this question, Jacob, because there was a I think you're referring to a preprint. There's a preprint from Brian Roth's lab where they have the 5-HT2A mouse line that they created, where they was questioning about whether some of these 2A receptors might be expressed in the Thywan neuron. Actually, we did some more experiment, and our results were a bit in conflict with Brian's result. When we stain for 2A antibody, we do find that 2A to be present at the Thywan neuron. And then also, there has been a couple of other groups that look at the Thywan neuron. They think most of that is likely to be the PT subtype. And if you look at the transcriptomics from the Allen, the PT subtype clearly have the 2A receptor. So I think from these two lines of evidence, we think the Thywan neuron does have the 2A receptors. So yeah, I think it remains to be clarified why I think Brian sees different things, but we think the 2A receptor are in the Thywan cells. Next question, have you looked at dendritic spine regeneration in layer 3 DLPFC? No, we have not. Well, first of all, I think rodent medial frontal cortex is quite different from the primate or human prefrontal cortex. If anything, I think the rodent doesn't have a prefrontal cortex, right? I would think the frontal cortex in the rodent correspond more to the anterior cingulate cortex of primates. And then for DLPFC, I'm not aware of primate work with psilocybin that have answered this question. Yeah. I think that will require basically a study in the species that have the DLPFC.
It looks like, yeah, another question from David. What are your thoughts on the recent pushback against the psilocybin trials conducted at Johns Hopkins, particularly on double blinding being insufficient as an experimental paradigm for collecting data in psychedelic research?
Yeah, I don't want to be specifically talking about another institution, but I thought actually the general criticism was not on the double blinding part, but more generally the field, and not specifically Johns Hopkins. I think many people actually tend to present these research to be more suggestive, as in when they present the research is not as sterile as one hope, where they bring in people maybe tend to already have some religious background or have certain expectation of these drugs, or maybe a drug user. I thought that was a criticism that's been around in the field. But from what I understand, people have changed. I think some of the current people directing this institute are aware of these issues, and it's being cleaned up. And the newer study, I have hope that it's much more regularized and done in a better way. Because I think, yes, the drug, yeah, I mean, some of these trials should be done, I think, in a more controlled fashion. I agree with that. Yeah.
Great.
And then Thomas Chow, I see. Do you have any localization and function of 2A receptor in the amygdala? I don't have a good answer for this. Amygdala is not my wheelhouse. I would point you to a very exciting study, an interesting study from Melissa Herman. I don't remember if it's published, but at least it's in preprint. But they look at 2A receptor in the amygdala. They're expressed in the amygdala. In our CFOS study, the BLA and I think the central amygdala lit up by psilocybin. So definitely there's CFOS in there. And Melissa did some photophotometry to look at that region in the context of threat and safety, I believe, and have some interesting finding. So I think it's well worth studying. It's not something my lab does, but I think it's a fascinating location to look at.
Chantal asks, how can you tell if increase in CFOS in a part of the brain is due to effects solely in that part versus effects helped along by connecting part to that part of the brain?
Yeah, we don't know. I think it definitely could be driven by other places. I mean, I think the CFOS, what you can know is that it's activity-dependent transcription, but whether that activity is due to the drug acting on those neurons directly, or is the drug acting on some other region that synapse onto those neurons, that's something that we don't know. I think the way to do that would be try to locally infuse these drugs in and then do the CFOS study. That's not something that we have done, and obviously very difficult for many brain regions. But if one want to know for a specific brain region, that would be the kind of research to do. In fact, I mentioned a little bit briefly about the dorsal raphe study from George Akaginian. One of the things I like a lot about his study is he actually did that part. He in addition to systemic administration, he also locally injected things like LSD into the dorsal raphe, which I don't see being done these days, but it's an important part of trying to understand what's going on.
Okay, great. In the interest of time, I'm going to kind of skip around a little bit. Ben asks regarding affected excited cells, has there been more depth in understanding the central nervous cells affected within the spinal cord also, i.e. beyond the brain?
Yeah, actually, I was just talking to a colleague at Yale who's very interested in looking at maybe spinal cord injury and regeneration, but I don't think much has been done. There are serotonin receptors out there, but I think, again, it's worth looking at. There's so many things, I think, but exciting. But I don't know much beyond that. Yeah.
Okay. Jabi asks, how do you interpret the head twitch response? Are there any comparable or related behavioral subjective effects in humans, in your opinion? And I also had personally a related question. I was wondering how you characterize the head twitch response. I saw you had linked some code there. So I was wondering about that as well.
Yeah, the second part first. The head twitch, we characterize two ways. One is with overhead video camera, like I show. The other is with ear tag magnet. So that's, initially started by Adam Halberstadt lab and then later on by Javier Gonzalez Mezo. We basically took those ideas, but then we put a very detailed parts list and assembly instruction on GitHub. I encourage anybody, and it's very cheap setup, so you can go and buy all the stuff and make your own. So I encourage you to check out that GitHub. And then how do you interpret head twitch response? I think you should interpret it very carefully and literally. Head twitch is just the head moving. It's a rodent thing. It doesn't happen in human. As far as I know, after human consuming psychedelics, they don't move, or at least not twitch the head. So it's a behavior that has no construct validity. So I think you should interpret it accordingly. Yeah. Yeah.
Yeah, for sure. I know that's a big question going on nowadays in rodent research. Okay, Javed asks, first says, thanks for the excellent talk. Two questions. One, there's quite a bit of variability in the behavioral responses to psilocybin. Do you expect this to be reflected at the molecular scale? Perhaps in variability of epigenomic predisposition to the drug? And then would you expect the same level of spine induction to be the same in an animal raised in a hyper-enriched environment?
Yeah. Actually, sorry, one more thought from the last question about the head twitch. What I actually would really like is a perceptual task for rodent, which I think is an opportunity for somebody to develop a visual type of task or other kind of sensory task that actually could more accurately capture that hallucinogenic effect. I haven't seen that, I think, but there are enough of these sophisticated perceptual tasks now in rodents where that might be possible to actually develop a task like that. That would be a big advance and a big positive thing for the field if such an assay exists, that we can move beyond the head twitch and have something that have construct validity. With regard to spine induction, yeah, I think it's an open question and very interesting question on whether these mechanisms are related to learning or different from learning. I think that's what you're trying to get at, or maybe environmental enrichment. How much of these spine formation mechanisms are the same, again, as learning or not, and whether they can maybe add on top of each other or maybe not. I think that's an open question. We have not tested it, but I think it's well worth asking and trying.
Okay, great. Well, it looks like we're at the top of the hour now, so I apologize to all the questions that we weren't able to answer. But thank you all so much for this wonderful discussion, and thank you also to Dr. Kwan for your fascinating talk. If students would like to learn more about your work or any work in this field or get in touch with you, where can they reach out?
You can email me directly. I'm quite responsive on email. You can also reach me on Twitter. You can message me. I'm happy to talk more. If I can't answer your question, particularly the one you asked here, yeah, feel free to reach out. I'll be happy to answer.
Great. Well, thank you so much, and we hope to keep in touch.
Definitely. Thank you.
Thanks.